Chapter 15, Microbial Mechanisms of Pathogenicity. Oh, I have gone through this PowerPoint and I have eliminated a lot of repetition. So only what is in this PowerPoint in lecture would be on the quiz. So when you're talking about pathogenicity, you're talking about the ability of the microbe to cause disease. And the virulence is the extent. So if something is very virulent, then it's much more dangerous than something that is less virulent. Avirulence would be a microbe that doesn't cause disease. Portals of entry. How can the pathogen get in the body? Mucous membranes. So if you inhale, let's say infectious droplets, then the mucous membranes of the nose and the mouth, respiratory tract can readily become portals of entry. The skin, if the skin has some sort of break in it, normally skin is a dead layer and it's also keratinized, so it's waterproof. And only if that is disrupted can a microbe, a pathogen enter. Parenteral would be injection. Okay, so the number of invading microbes, in other words, the infectious dose. And ID15 means for 50% of the test population. And lethal dose is obviously 50% of the test population that when they are infected dies. So how much or how high a level of the microbe or its pathogen is needed to kill? Right. So one of the things that can make a microbe pathogenic or more virulent is the ability to adhere to body surfaces inside organs and organ systems. So a fimbriae from, there are E. coli that are highly pathogenic and can, like hemorrhagic E. coli that has killed young children in the United States. So the problem is that these short little hairs or fimbriae on the E. coli make it very easy. Think of them like Velcro, that they can stick to the sides of the lower gastrointestinal tract, and it's very difficult to remove. And then once they are stuck there, they then can make their toxins and kill. Strep pyogenes, the causative agent for septic sore throat, can also reside and stick. Because remember, what you do is you can drink water, you can eat food. I mean, you can uh, end up in inhaling certain things that could dislodge these pathogens. But if they have a way of sticking, and it's very difficult to dislodge them. So there's an M protein on the surface of strep pyogenes that makes it very difficult to dislodge, and so it can establish a foci of infection very quickly. All right, enzymes that can be made by pathogens. I just have three: coagulase, collagenase, and IgA protease. And they are going to be my poster children for how enzymes that are made by pathogens can bypass or spread or destroy immune responses, which of course makes them more virulent and more difficult to get rid of. So coagulase means to make a blood clot. And you'd say, well, why would a pathogen? And staph and streps can do this very easily. Well, if you have a staph infection and it gets in the blood, then it's able to make this coagulase and spin a blood clot. Now, they actually hide inside the blood clot, which makes it very difficult for the immune system to detect them because they're hiding within the blood clot. And besides evading the immune system, if you're in a blood clot, you can spread all over the body in seconds. Collagenase. A lot of organs have collagen, and that's extracellular matrix, and it's there to bind things, and it's also there to prevent pathogens from spreading from one tissue, from one organ to another organ. So, if you're a pathogen and you're highly virulent and you're in one part of the body and you run out of nutrients, you would like to spread to another part of the body where there are more nutrients. And so they can produce collagenase, and staff can do this quite readily, and it's going to hydrolyze. It's going to basically digest the collagen so that it can very readily spread from organ to organ. Now, IgA is an antibody of body secretion. So antibodies are great for binding pathogens, and that's going to start them down the road of being destroyed. IgA is made and found in mucus, tears, sweat, and that's there to protect you against pathogens that could enter the eyes, enter the skin, enter the upper respiratory tract. So if it is a pathogen that can make these proteases, then they're going to evade immune attack on those area or in those areas of the body that make IgA. So you can develop an eye infection because IgA is normally found in tears 24/7. So the pathogen makes the protease to destroy the IgA antibodies to help the pathogen evade destruction. Toxins. A highly virulent organism very often can make toxins, poisons. And the poisons or the toxins are what make the pathogen so lethal. So a toxin is any substance. It could be a lipid, it could be a carbohydrate, it could be a protein. That's going to contribute to the pathogenicity and virulence of the microbe. Toxigenicity is the ability to produce the toxin. Toxemia is the presence of the toxins in the person's blood. And that's the important thing, because if it gets in the blood, it can be disseminated and become systemic in no time flat. A toxoid is our ability to address that. So very often it's included in a vaccination, and I'm going to give you an example down here. So it's an inactivated toxin. Vaccines are a way of trying to fool the immune system into making an immune response without the person getting sick. So let's go to this example of an antitoxin. Antibodies against a specific toxin. So when, let's say that you think you've been exposed to food poisoning, that is botulism, and you are able to present to the ER and get an antitoxin shot. This can also happen when you're trying to avoid Clostridium perfringens, which causes gangrene, or other types of pathogens. So let's just stick with the botulism. So the botulism antitoxin is injected into people exposed to Clostridium botulinum in order to immediately neutralize the botulism toxin. Clostridium is a genus that is just flooded with all kinds of horrible pathogens that produce horrible toxins, and the Clostridium itself is not that pathogenic. What is pathogenic about Clostridium, whether it's Clostridium perfringens or botulinum, etc., tetanus, Clostridium tetani, is the toxin they produce. So Clostridium tetani produces tetanus toxin, which can produce lockjaw, and botulism can produce a neurotoxin that can kill. Perfringens is able to make a toxin that can liquefy flesh. And in all cases, if you know that you're exposed to it early on, you can get an antitoxin. And an antitoxin is an antibody that will specifically bind to that toxin, sweep it out of your body. You either pee or poo out of your body, and then you're fine. And then they can treat you with an antibiotic that can get rid of the Clostridium, and you live happily ever after. So that is our ability to produce antitoxins, and then in this case, antitoxins are an antibody, or if you have time. See, vaccines take about 2-3 weeks, up to 6 weeks, to actually establish protection. So a toxoid, it's great to inactivate a toxin and use it in a vaccine, but you'd be long dead if that was Clostridium botulinum. So if you know that you've been exposed, an antitoxin can nail it within minutes. Okay, there are two broad categories of toxins: endo, E-N-D-O, endotoxins, and exotoxins. So endotoxins mean that the toxin is within the bacteria itself while it's alive. And when it's alive, the endotoxin cannot hurt you. It's when you kill the bacteria that has the endotoxin and the death of the bacteria will release it. Or something happens that the endotoxin is released. Very often it has to do with when you kill the bacteria that contain the endotoxin. That's the easiest way to release it. Endotoxins are about gram-negative bacteria, and the endotoxin is usually the outer lipid or the lipopolysaccharide, the LPS layer. All I really want you to know is endotoxins an example is lipid A of this gram-negative bacteria. And it's liberated when the bacteria die and the cell wall breaks apart. So the lipid is actually part of the gram-negative cell wall. So you kill the gram-negative bacteria, it releases that LPS lipid A, and then that starts you down the merry road of toxicity. So endotoxins, source, not all gram-negatives, but the pathogens. Metabolic product, it's present in the LPS layer, lipopolysaccharide layer of the outer membrane. The chemistry, it's a lipid, it's a fat. Does it cause a fever? Yes. Endotoxins can cause fevers. Can it be neutralized by an antitoxin? No. An antitoxin is a toxin that's circulating in your body when the organism is alive. So that's a different kettle of fish. And that brings us to, exotoxins. To exit, to be released, to exit in this case, the bacteria. So gram-positive bacteria that are virulent, that are pathogenic, can produce and release their own toxins while they are alive. If you kill the exotoxin, if you kill the bacteria making the exotoxin, then the bacteria will stop producing it. But if there is still exotoxin in the body, you can still have extreme effects due to the exotoxin. So here is a gram-positive organism that is virulent because it produces these exotoxins while it is still alive, has to be alive in order to make it. So here we have exotoxins are produced inside mostly gram-positive bacteria as part of their growth and metabolism. They are secreted and released, and then away we go. So exotoxins are produced mainly by gram-positive pathogens, by product of a growing cell. It is a protein, does not produce fever, and can be neutralized by an antitoxin. As you can tell, it becomes separate from the bacteria. So antibodies binding to the toxins, which is really what makes the pathogen dangerous, you neutralize it, then of course you hit it with an antibiotic to get rid of the gram-positive organism, and you're all set. There is something called superantigens. They are extremely virulent, very toxic. Don't ever want to have that happen. And that's why they're called super antigens. Just mean those molecules, usually on the surface of a pathogenic microbe, pathogenic bacteria, that are seen as foreign or different by the immune system. The immune system sees these outer components as foreign or different from you, yourself. They're non-self, and the immune system will then mount a response to get rid of them. Super means that they're super dangerous. So, and what makes them super dangerous is the intense immune response. So certain really virulent strains of staph infections, if they get systemic, it's just going to be a matter of time. The person either presents and gets treated intensely very quickly after infection, or death is something that's not uncommon. So superantigens produced by deadly virulent staph that becomes systemic in the body, usually via the bloodstream, can cause fever, nausea, vomiting, diarrhea, shock, and death. So they're kind of like in a class of their own. So here are some that are exotoxins that are problematic. So diphtheria, we vaccinate our children against diphtheria because it can be, well, it was a killer. And Corynebacterium diphtheriae are the organism that produce the diphtheria toxin, and it inhibits protein synthesis. And now I'm not going to get into lysogenic conversion, so we can forget about that. That's a separate discussion, and just not going to cover it on the quiz. Strep pyogenes, that is going to give you septic sore throat because of an exotoxin that is erythrogenic, meaning that it destroys red blood cells. Clostridium botulinum, fatal food poisoning. It produces a neurotoxin. Clostridium tetani, that stepping on a rusty nail, better make sure that your tetanus shot is up to date. And that can kill, that can kill by a neurotoxin that tetani produces that creates lockjaw. Vibrio cholera, that produces a dysentery that can be deadly and give a person cholera. And that is an enterotoxin that can produce such massive diarrhea that the person can become so dehydrated that they die quickly. Staph aureus, and that produces a lot of different toxins, but it can produce an enterotoxin that's a super antigen and is quite deadly. Portals of exit. This is the last slide. So with portals of exit, how can a pathogen inside of you become infectious to other people? So you can have respiratory tract, people cough, people sneeze. You can have the GI tract, saliva, feces. And so if there's not a lot of sanitation, if you have water that's contaminated and people drink it, then they can get GI tract pathogens. Genital urinary, urine or vaginal secretions. So we can also have portals of exit in the skin where the person might have a boil and it ruptures, and that's infectious. And blood, that tends to be needles and syringes, and the other biting arthropods. Hey, summertime, the mosquitoes, the ticks, all those lovely blood-sucking creatures that can carry and transmit disease. So